The first video in this lecture series on metabolism covered fundamental principles. If you have not already, go back and watch that video. It covers all sorts of concepts that are necessary to understand what we cover in this video. Our focus here will be on fermentation. Before we begin, let's take a little bit of a step back and think about catabolic processes in general. In reality, metabolism is pretty simple. There are only three major types of catabolic processes on Earth. Two classes get their energy from the oxidation of chemicals, while photosynthetic microbes get their energy from light. These classes are photolithoautotrophs. They get their energy from light, their electrons from inorganic sources, and their carbon is often from CO2. Second, chemoheteroorganotrophs. They get their energy from the oxidation of organic compounds. They get their electrons from organic compounds, and they get their carbon from organic compounds. The third class is chemolithoautotrophs. They oxidize inorganic chemicals for energy. Inorganic. They attain their electrons from inorganic compounds, and they get their carbon from carbon dioxide. Now, there are other types of metabolism known on Earth, but these are probably the biggest ones. We will cover the processes they use in depth in the next three lectures, and we'll talk more about these classifications in the lectures on nutrition. The energy sources can be light or chemicals. From there, the cell uses just two systems to generate energy. So they capture that energy from light or chemicals, and then they generate common energy products using two systems. First is substrate level phosphorylation, SLP. It synthesizes ATP from ADP, and this is directly linked to the breakdown of high energy organic substrates. The second method is membrane system phosphorylation. In this system, high energy electrons are extracted from the energy source and passed through a series of electron carriers. The cell uses the energy released from these redox reactions to pump protons across the membrane. That resulting proton gradient is converted into ATP for the cell. We will go through the specifics of SLP and membrane system phosphorylation as we cover these processes. This lecture is going to focus on fermentation, which uses substrate level phosphorylation. We now move on to look at some of the more common catabolic pathways. Glycolytic pathways are those that degrade sugars. People also use this term for the initial catabolism of glucose and call it glycolysis. We begin by describing the breakdown of sugars to generate energy. This pathway is present in many different organisms, including you. It's also present in microorganisms, but it's not their only method. We start here because it's a great jumping off point for you because you are likely familiar with it from other classes. Common pathways that are used are the Embden Meyerhoff pathway, EMP, and the Enter-Deuteroff pathway, EDP. EMP is found in many different bacteria and in eukaryotes. This is likely the one that was covered in your biology class. EDP is used by other bacteria, typically strictly aerobic bacteria such as Pseudomonas. I will go through the Embden Meyerhoff pathway quickly as you have seen it multiple times. Glucose is a chemically stable molecule and will not undergo reactions easily. Thus, it is necessary to activate the molecule. During activation, phosphates are added to destabilize glucose. Glucose is converted in two steps into fructose 1,6-diphosphate. Fun fact, German scientists began to investigate this process of glucose catabolism to understand what was happening during beer fermentation. Zymology, the study of fermentation, gave us the term enzyme, which has at its root zymology. After activation comes splitting of the 6-carbon molecule. Aldolase splits the activated fructose 1,6-diphosphate into diglyceraldehyde 3-phosphate, which I will call GAP, and dihydroxyacetone phosphate. Activation and then splitting is a common pattern you will see in the catabolism of carbon compounds and others. GAP then proceeds down the pathway. Triisoisomerase then converts dihydroxyacetone phosphate into GAP, so it can also be catabolized. Isomerase can interconvert between the two 3-carbon sugars, and in fact the equilibrium of this process is about equal concentrations of both dihydroxyacetone phosphate and GAP. The cell drives the reaction towards GAP by continually moving it for use further down the pathway. This configuration is the example of the cell manipulating the equilibrium of reaction to drive it in the desired direction. In the next 5 steps, the cell converts GAP to pyruvate. In the process, it generates 4 ATP from the 2 GAP molecules. You have seen this all before, but I want to draw your attention to the first step here, catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It adds a phosphate to GAP, oxidizes the molecule, and does not require energy input. This is the heart of glycolysis and where it derives its energy. The ATP generated in this reaction is generated by substrate-level phosphorylation. The phosphate is directly transferred from the organic chemical to ADP. Now, let's look at a different pathway you probably haven't seen before. The Entner-Duderoff pathway begins in a similar manner to EMP, but it then diverges. After making glucose 6-phosphate, it oxidizes it to 6-phosphogluconic acid and then proceeds to those different steps. The end result is still the same, pyruvate and ATP, but the net is 1 ATP and 2 reducing equivalents of NADH. Aerobic respirers will use this pathway because they get an extra NADH, which they can use in their electron transport chain that we'll cover later. End product formation. The end products of glycolysis are pyruvate and NADH. Pyruvate is used as a carbon source. Some NADH is used in biosynthetic reactions, but there is an excess of NADH. For organisms that grow by fermentation, meaning that they do not use an electron transport chain, they need to dump these electrons and regenerate NAD+, so that they can continue with the pathway. Several of the solutions the microorganisms have come up with are things humans enjoy, as you will see. I mentioned fermentation in the previous slide, so let's define it. Fermentation is an energy-yielding process whereby organic molecules serve as both electron donors and electron acceptors. There are several themes in most fermentations. First, there are many types of fermentation, and we will only cover a few of them. Second, NAD+ is almost always reduced to NADH during the catabolic process. The NADH then has to be converted back to NAD+. Pyruvate is an important intermediate. Some more general concepts. Fourth, energy in fermentation is derived from substrate-level phosphorylation. Reducing power is in abundance and needs to be gotten rid of, as we described previously. Fifth, energy yields are low, especially compared to other types of metabolism. Organisms who ferment take advantage of environments where fermentation is the only possibility, and oxygen is absent. Typical substrates are sugars and amino acids, while typical products can be organic acids, alcohols, and gases such as hydrogen and CO2. Here are some important fermentations. First of all, alcoholic fermentations, which are important in alcoholic beverage productions. Lactic fermentations used in many food products such as yogurt, cheese, and kombucha. There can be homofermentations and there can be heterofermentations. And what that means is some homofermentations will produce lactic acid only. Heterofermentations will produce other products, as you'll see. It's also important in our muscles. When we use them at high levels at activity, our muscles will go anaerobic and we will produce lactic acid. Finally, propionic fermentations, such as those by Propionibacterium, as in Swiss cheese and others. Let's examine a few of them in detail. Alcoholic fermentations. Alcoholic fermentations are a simple two-step reaction. First, decarboxyling pyruvate to form acetaldehyde. This is then reduced by dumping electrons from NADH into ethanol, thus regenerating NAD+. Yeast and Zymomonas mobilis are two prominent bacteria that carry out this fermentation. There is some interest in making biofuels from fermentation. This is a good point to ask, why should you care about fermentations at all? One good reason is beer, even if you don't drink beer. Brewing beer is a huge industry. There are over 8,000 breweries nationwide. 883,648 barrels of beer were made in Wisconsin, and that's from the U.S. Census data in 2022. At least 205 commercial breweries are present in Wisconsin. As far back as 6000 BC, Sumerian civilization was brewing beer. Sumeria was in present-day Iraq. It was thought that people began to group together to grow barley and make beer. A prayer to the goddess Ninkasi, known as the hymn to Ninkasi, serves as both a prayer as well as a method of remembering the recipe for beer in a culture with few literate people. Ancient Chinese civilizations were brewing kuai, a beer-like substance. So, lots of civilizations have been brewing beer for literally tens of thousands of years. Brewing involves 4 ingredients: malted barley, water, hops, and yeast. These are combined in a complex process that results in the conversion of extraction sugar in barley being fermented by yeast into alcohol. Brewing begins by harvesting the barley and soaking it in water. This causes the grain to germinate, which activates amylases in the grain. During mashing, the amylases degrade the starch present into sugars, such as maltose, sucrose, and glucose. The choice of grain, how the malt is roasted, and how it is mashed is a common point of differentiation for beer. The mash is then put in a kettle and boiled with the addition of hops. Hops give the wort, as it is now called, antiseptic properties that prevent it from spoiling. After the boil, the wort is cooled and yeast are added to ferment the sugars into alcohol using the glycolytic pathway. Finally, the beer is aged and then packaged. Knowing the biochemistry of the fermentation can help you diagnose problems. For example, acetaldehyde, an intermediate in ethanol fermentation, as shown in the pathway, has a green apple taste. It can accumulate when the ethanol concentration gets too high. This can happen if your starting wort has too much sugar and that intermediate and that green apple taste is undesirable. We now move on to lactic fermentation. Note, do not confuse lactic acid fermentation with lactose catabolism. That is a common misconception that students have. This is the formation of lactic acid from fermenting sugars. While some lactic acid bacteria can use lactose, they can also use other sugars, such as glucose, fructose, and galactose. The product is always the same, lactic acid. In homofermentation, there is one end product, lactic acid. Lactic acid bacteria use the glycolytic pathway just like alcoholic fermentation. However, pyruvate is reduced to lactic acid in a one-step reaction. 2 ATP are added in the early steps of glycolysis, and 4 ATP are generated for a net gain of 2 ATP. This fermentation uses a large amount of substrate to generate a relatively small amount of ATP. Despite the low yield of the pathway, many bacteria have been successful using it, including Streptococcus, Lactococcus, and some lactobacilli. Heterofermentation is another way that some bacteria ferment glucose. The yield is only 1 ATP per glucose, with lactic acid, carbon dioxide, and ethanol being end products. Even with its small yield, leuconostoc and some lactobacillus use homofermentation as their form of metabolism. Lactic acid bacteria have a wide utility in many different types of fermentations. The cheese business always comes to mind in Wisconsin, eh? Lactic acid bacteria are the ones that carry out the first steps of the fermentation. Raw milk is the substrate. The main components of cow's milk are fat, protein, and lactose, and it has a pH of about 6.6. This is an ideal environment for lactic acid bacteria, which will happily ferment the milk under the right conditions. Cheese is a favorite food worldwide, with about 21 million tons of cheese consumed annually. There are over 1,800 types of cheese, but they all rely on the action of lactic acid bacteria to ferment the lactose in milk to lactic acid. The process begins with the pasteurization, or heating, of raw milk to kill pathogens. An added starter culture then ferments the milk, producing lactic acid and lowering the pH. The addition of rennet, which is mostly proteases, and the low pH causes the milk to curdle, forming curds. For fresh cheeses such as cottage cheese, mozzarella, or feta cheese, that is the end of the process. They are packaged and sold to the consumer. For hard cheeses, squeezing of the curd and pressing into molds removes water. This excess water is whey and is the source of whey protein. Aging of the cheese for weeks to years gives it distinct properties, and the methods of aging, how much salt, the addition of starter cultures, the temperature of the room, etc., dictate the kind of cheese that ends up being produced. After aging, the cheese is then removed from the molds, packaged, and sold to consumers. Fermentations also occur across the world, and another example of lactic acid bacteria fermentation is kimchi. Kimchi is the fermentation of cabbage and other vegetables by lactic acid bacteria and coliforms. Coliforms are another type of bacteria we will cover in more detail later. Lactic acid is the major product of these fermentations and gives the finished product its flavor and texture. Kimchi history. Kimchi is an ancient practice, being referenced more than 2,500 years ago in a Chinese poetry book. There are hundreds of known varieties. Its original form was just cabbage and beef stock, but spices were introduced when societies in Asia started trade with European countries. Kimchi is a staple of Korean cuisine. Most Korean families will have some kimchi at every meal. The most popular type is baichu, using Napa cabbage, and that can be seen in the upper left. Ingredients for kimchi depend upon the recipe, and this can vary. Kimchi involves brining the vegetables in salt water, which causes nutrients to flow out of the vegetables and creates a rich medium for microorganisms to grow. Kimchi is a natural fermentation, meaning that no starter culture is added. In beer brewing and in cheese making, a starter culture is added. The microbes that ferment kimchi come in on the ingredients. The progression of fermentation is a three-step process. First, coliforms such as Klebsiella or Enterobacter species ferment the available sugar to acidic products and drop the pH to about 5. At this pH, leuconostoc takes over the fermentation because the coliforms can no longer grow. It is a heterofermentative lactic acid bacteria and produces some gas. Below pH of about 4.5, it can no longer ferment. At this point, the third stage of fermentation is carried out by homofermentative lactobacillus, who produce more lactic acid and drop the pH to 4. At this pH, nothing can grow, and the high acid concentration has a preservative effect. You can see in this figure the progression of fermentation in this kimchi or cabbage product. Now, the actual time it takes varies depending on the recipe and the time frame, but the progression is the same. Coliforms, leuconostoc, then lactobacillus. We end this lecture on fermentation by describing the fermentation of chocolate. You didn't know chocolate was a fermented product? It is. Chocolate has been described as the food of the gods because of its appeal to so many people. It is another natural fermentation where the microbes living on the cacao pods do the fermentation. Chocolate is big business, with over 2.5 billion kilos of it being produced each year. Chocolate has a number of psychoactive products: anandamide, structure A, and theobromine, a stimulant, structure B. Anandamide is a chemical manufactured in the brain that is associated with a blissful feeling and is generated from running, yoga, and meditation. It is the chemical basis of the runner's high. Chocolate also has this compound and can generate similar feelings in some people. Theobromine is a stimulant that has a milder kick, but more long-lasting than caffeine. It also improves cognitive function, relaxes smooth muscles, and dilates blood vessels, among other effects. No wonder people love chocolate so much. So where does the fermentation come in? Well, first of all, chocolate fermentation is a complex process. What you didn't know is that chocolate production involves a microbial fermentation. And this microbial fermentation is a complex, multi-phase fermentation. Cacao pods have seeds that are covered in mucilage. The mucilage is mostly carbohydrate. Farmers will take the pods, break them open, and spread them out into shallow boxes that allow aeration and easy mixing. During fermentation, there is a complex succession of microbes. Early in chocolate fermentation, yeast ferment the sugars to ethanol, followed by lactic acid bacteria producing lactic acid. After 24 hours, acetic acid and bacterial populations increase. Temperatures increase up to 50 degrees, and at this time spore formers begin to appear, and non-spore formers are decreased because of the higher temperatures. Fermentation ends around day 7. If left too long, molds may begin to grow and produce off flavors. I hope this journey through some examples of fermented foods has made you aware of how important this microbial activity is to humans. I will finish this lecture by pointing out that we have only scratched the surface of fermented foods. There are many more we did not talk about, each with unique methods and some with unusual microbes. I show them in this table. However, this is information for fun only. I will not be requiring you to memorize this table and be able to regurgitate it back to me. This ends the lecture on fermentation. Our next topic in the next lecture will be on respiration.